All right, so today we're going to do a deep dive on something pretty incredible. CRISPR-Cas9. You've probably heard of it. It's this gene editing tech that's been making headlines, and for good reason. I mean, we're basically talking about molecular scissors that can cut and paste the code of life itself.
It's pretty mind-blowing. And the thing is, this isn't just science fiction anymore. We're talking real-world applications here, like potentially curing genetic diseases, fighting cancer, even engineering climate-resistant crops.
It really sounds like something out of a sci-fi movie, but it's real, and it's already having a huge impact.
Oh, absolutely. I mean, the fact that the discovery of CRISPR won the Nobel Prize tells you just how groundbreaking this technology really is.
Right. So let's dive into the history of CRISPR. Where did this all begin?
Well, it's kind of a funny story, actually. It all started in the most unexpected of places, bacteria.
Bacteria?
Yep. Back in the 1980s, scientists discovered these strange repeating sequences in bacterial DNA, but they had no idea what they were for.
So it was like a scientific mystery.
Exactly. It took years of research to finally crack the code. And what they found was that these sequences were actually part of a bacterial defense system.
A defense system?
Yeah, it's a way for bacteria to remember and fight off viruses that have infected them before.
So it's like a bacterial immune system.
Exactly. It's
Yeah.
like bacteria have this genetic memory of their enemies.
That's incredible. But how did we go from this bacterial defense mechanism to a tool that can edit genes in any organism?
That's where the work of Jennifer Doudna and Emmanuel Charpentier comes in. In 2012, they showed that you could actually harness this system to edit genes with incredible precision, and that was a total game changer.
So how does it actually work?
Well, it's like a molecular GPS system.
A GPS system?
Yeah. You design a custom RNA molecule. This RNA acts as a guide. It tells the Cas9 protein where to go in the DNA.
So the RNA is like the address?
Exactly. And the Cas9 protein, that's our molecular scissors, it goes in and cuts the DNA at that precise location.
Wow. And once you make that cut, what happens next?
That's when things get really interesting. The cell's natural repair mechanisms kick in.
So the cell tries to fix the damage.
Exactly. And there are a couple of different ways it can do that.
Okay, so let's talk about those repair mechanisms.
Yeah.
Because that's really key to how the gene editing actually works.
Right. So one way the cell can repair the DNA is through a process called NHEJ. That stands for non-homologous end. Joining, I know, right? But basically, it's like a quick patch job. It just sticks the ends of the DNA back together.
And what's the other way?
The other way is called HDR, homology-directed repair. This is a more precise repair mechanism. It uses a template to guide the repair.
So it's like having a blueprint for the repair.
Exactly.
So depending on which repair mechanism the cell uses, you can get different outcomes.
Right. With NHEJ, you can disrupt genes, which can be useful if you want to knock out a gene's function.
And with HDR.
With HDR, you can actually insert new DNA sequences.
So you can modify genes.
Precise.
Now, we've talked a lot about the potential of CRISPR to cure diseases, but I'm curious, what would a world without these genetic conditions actually look like?
That's a great question. It's really hard to even imagine, but I think it would be a profoundly different world. I mean, think about diseases like cystic fibrosis or muscular dystrophy. If we could just erase those from existence.
It would be life-changing for so many people.
Absolutely. Not just for the individuals and families affected, but also for healthcare systems and society as a whole.
That's a really good point. It would free up so many resources.
Exactly. We could focus on other areas of healthcare, preventative measures, new innovations.
It's like rewriting the future of healthcare.
It really is.
But of course, with such power comes great responsibility. How do we make sure that this technology is used ethically and that it doesn't create new inequalities?
That's the million-dollar question. We need to have open and honest conversations about the ethical implications of CRISPR. We need to involve everyone in this discussion. Scientists, ethicists, policymakers, the public.
It's a complex issue with no easy answers.
Definitely.
Okay. So we've talked about CRISPR's potential to cure diseases. But what about its role in fighting cancer?
Well, this is another area where CRISPR is showing incredible promise. One approach is to use CRISPR to engineer immune cells, like T cells, to make them better at recognizing and destroying cancer cells.
So it's like giving our immune systems a boost.
Exactly.
That's pretty amazing.
It is, and it's already showing promising results in clinical trials.
Now, what about other diseases caused by viruses, like HIV?
Yeah, that's a really exciting area of research. Scientists are exploring ways to use CRISPR to target and disable viral DNA, essentially trying to eliminate the virus from the body.
It sounds like something out of a science fiction movie.
I know, right? But it could become a reality.
That would be incredible.
It would. It would be a game changer for so many people.
So we've talked about curing diseases, fighting cancer, but CRISPR is also revolutionizing the way we diagnose diseases.
It is. Remember those repeating sequences in bacteria we talked about earlier? Well, it turns out they're incredibly useful for detecting specific DNA or RNA sequences.
Oh, interesting.
So scientists have developed CRISPR-based diagnostic tools like Sherlock and Detector. These tools can rapidly identify pathogens, including viruses like COVID-19.
Wow. So we're talking about faster and more accurate diagnoses.
Exactly. And this has huge implications for public health.
Absolutely.
Yeah.
Now, CRISPR is also having a major impact on agriculture.
It is. It has the potential to revolutionize the way we grow food.
How so?
Well, imagine crops that are more resistant to pests and diseases, crops that can thrive in harsh climates, crops that produce higher yields.
So we're talking about engineering crops that can help feed a growing population.
Precisely.
That's really important, especially with the challenges we're facing in terms of food security.
Absolutely. And it's not just about crops. We can also use CRISPR to enhance livestock, making them more resistant to diseases and more productive.
So CRISPR has the potential to make a real difference in global food security.
It really does.
But now let's talk about the elephant in the room, the potential risks and ethical concerns surrounding CRISPR.
Right. Because with such a powerful technology, it's important to be aware of the potential downsides.
Absolutely. One concern I've heard is the potential for unintended consequences.
That's a valid concern.
Yep.
One potential risk is off-target edits, where the Cas9 protein cuts the DNA at the wrong place.
And could that be dangerous?
It could be. It could lead to unforeseen consequences.
So are these off-target edits a major problem with CRISPR?
Well, scientists are working hard to improve the precision of CRISPR. Newer versions of Cas9 are much more accurate and less likely to cause off-target edits.
That's good to hear. But what about the ethical implications of using CRISPR to alter the human germline, making changes to DNA that could be passed down to future generations?
That's a very controversial topic. There are a lot of ethical concerns about germline editing. Some people believe that it's wrong to tamper with the human genome in this way.
And what's the scientific community's stance on this?
Well, there's a general consensus that we need to proceed with caution. We need to have a lot more discussion and debate about the ethical implications before we start editing the human germline.
It's a complex issue.
It is, and it's one that we need to approach with a lot of thought and care.
Okay, so we've covered a lot of ground today.
Yeah.
But I think it's clear that CRISPR is a truly revolutionary technology.
It is. It has the potential to change the world in so many ways.
It's incredible to think that we can now harness the power of a bacterial defense system to edit the very code of life.
It's pretty mind-blowing.
But as we've discussed, this power comes with great responsibility.
Absolutely.
We need to be mindful of the ethical implications of CRISPR
Agreed.
and make sure that it's used for the benefit of all humankind.
Exactly.
Okay, so we've talked about how CRISPR can cure diseases and fight cancer and even revolutionize agriculture. But what about the future? What's next for CRISPR? Like, what are some of the more out there applications?
Well, one area that I think is incredibly exciting is precision medicine. Imagine a future where treatments are tailored to your specific genes. Like doctors could use CRISPR to modify your cells or even correct genetic defects before they cause any problems.
So it's like personalized medicine on a whole new level.
Exactly. And the thing is, the CRISPR toolbox is constantly growing. Scientists are discovering new CRISPR enzymes all the time, like Cas12, Cas13, and Cas-X. These enzymes are even more precise than Cas9, and some of them can even target RNA.
RNA editing. That sounds pretty advanced.
It is. It basically allows us to make changes to the RNA without affecting the DNA.
So it's like a temporary fix.
Yeah. And that could be really useful for certain conditions.
That's fascinating. Now, we've talked about CRISPR's potential in medicine and agriculture, but what about its impact on the environment?
Well, CRISPR could actually play a crucial role in tackling climate change and pollution.
Really? How so?
Well, we've already talked about engineering climate-resistant crops, but you could also use CRISPR to engineer bacteria that can break down pollutants, or clean up oil spills, or even capture carbon from the atmosphere.
That's incredible. So CRISPR could help us create a more sustainable future.
Exactly.
Now, with all this talk about pushing the boundaries of science, there are bound to be some ethical concerns.
Absolutely.
What about the idea of using CRISPR for human enhancement? Like, could we use it to make ourselves smarter or stronger or even live longer?
Yeah, that's a really tricky question. The idea of enhancing human capabilities is fascinating, but it also raises a lot of ethical dilemmas. Like, Who would have access to these enhancements? Would it create even more inequality in society? And what would be the long-term consequences for humanity?
Yeah, it's like something straight out of a sci-fi movie.
I know, right?
But these are questions that we need to start thinking of now.
Definitely. Because the technology is advancing rapidly.
So let's bring it back to the present for a moment. We've talked about all these amazing possibilities. But what's the current status of CRISPR? Is it actually being used to treat people right now?
It is. In fact, there have already been some pretty remarkable successes in clinical trials, using CRISPR to treat blood disorders like sickle cell anemia and beta thalassemia.
Wow. So CRISPR is already making a real difference in people's lives.
It is, and the field is moving so quickly. There's a lot of research going on to improve delivery systems and make CRISPR even more precise, and to expand its use to treat even more diseases.
It really feels like we're witnessing a scientific revolution in action.
I think so too.
Now, you mentioned earlier that CRISPR could be integrated with other technologies.
Yeah.
What might that look like? And what kind of advancements could we see?
Well, one really exciting possibility is combining CRISPR with artificial intelligence.
CRISPR and AI.
Yeah, AI could help us design even more effective guide RNAs. And it could help us predict potential off-target effects and even personalized treatments based on a patient's unique genetic profile.
So it's like CRISPR with a brain.
Exactly.
That's amazing.
And it goes beyond AI. We could also combine CRISPR with advanced biomaterials to create targeted drug delivery systems, or even engineer tissues and organs for transplantation.
Wow.
And then there's nanotechnology. We could use nanoparticles to deliver CRISPR components directly to specific cells, which would increase efficiency and reduce side effects.
With CRISPR plus AI plus nanotechnology plus biomaterials, the possibilities are endless.
I know, it's pretty mind-blowing.
It really is.
CRISPR is going to revolutionize medicine and agriculture and biotechnology in ways we can't even imagine yet.
And we're just getting started.
Exactly.
Wow, it's amazing to think about how our CRISPR has come in such a short time. From those bacterial defense systems to all these incredible possibilities. I mean, it's clear that CRISPR is a complete game changer.
It really is it's incredible. It has the potential to solve some of the biggest challenges facing humanity, like curing those devastating diseases and making sure everyone has enough food to eat.
Right.
But as we've been talking about, this power comes with a lot of responsibility.
For sure.
We have to be really careful about how we use this technology.
Absolutely. I mean, it's not just a scientific breakthrough, it's a societal one, too.
Yeah.
CRISPR could completely change our world. And it's up to all of us to make sure we're using it wisely.
Couldn't agree more. We have to keep talking about the ethical implications and really think about the potential benefits and risks. It's going to impact everyone, so everyone needs to have a say.
So as we wrap up this deep dive on CRISPR, I'm feeling a mix of awe and cautious optimism. It has so much potential.
Yeah.
But it also comes with some big ethical challenges.
Definitely.
It's like it's up to us to guide this powerful tool in the right direction.
Well said. It's a tool that could change the world for the better. But we have to be smart about how we use it.
Well said. And to all of you listening out there, remember you have a role to play in shaping the future of CRISPR. Stay informed, join the conversation, and ask those tough questions. Because the future of CRISPR, and maybe even the future of humanity, is in our hands. Thanks for joining us on this deep dive into the world of CRISPR-Cas9